Hello, I'm Chris McMurran from the University of Cambridge, and this is a video abstract for our paper, Exploratory Analysis of Biological Age Measures in a Remyelination Clinical Trial, published in Brain Communications. This was a collaborative project with Dr. Declan Chard and colleagues at the Queen Square Multiple Sclerosis Centre at University College London. In people with multiple sclerosis, the body's own immune system attacks myelin sheaths, which are the insulating material around nerve cells. The brain has an inbuilt mechanism to repair this damage, known as remyelination, and successful remyelination might help prevent subsequent axon loss and disability. But data from preclinical studies, and now also people with multiple sclerosis, shows that this repair process becomes less effective as we age. Is this an inevitable consequence of aging, or could we in theory intervene to boost brain repair across the lifespan? Age is normally measured in years since birth, or chronological age, but aging can look very different between different people. These observed differences in aging have given rise to the notion of a biological age, which is different to one's chronological age and, unlike chronological age, could in theory be modified. In this project, we used existing clinical trial data to generate two measures of biological age, one using MRI brain data and the other generated from standard clinical blood tests. And we explored the relationship between these biological age measures and brain repair compared to chronological age alone. We used data from the CCMR1 trial, which tested a drug called bexarotene for its effect on remyelination markers in 49 people with stable relapsing remitting multiple sclerosis. Although bexarotene had a number of side effects, it also caused significant improvements in several markers of remyelination. This was measured in two ways. Firstly, the speed of transmission of the signal along the optic nerve to the brain, or VEP latency, and secondly, an MRI measure called MTR. In previous work, we showed that some of these beneficial effects tail off with chronological age, particularly around the 5th decade of life. Consistent with existing literature, we showed that this cohort of people with MS had an MRI brain age on average 10 years older than expected for their chronological age, albeit with substantial variability between individuals. The average blood biological age was very close to their chronological age. During the 6-month trial, the MRI brain age of people taking the placebo increased by a few months as expected. Those taking bexarotene to promote remyelination had a significant reduction in their MRI brain age. In this group, MRI brain age appeared almost 1 year younger at the end compared to the beginning of the study, despite 6 months of time passing. In linear mixed models, this apparent rejuvenating effect of bexarotene on MRI brain age was associated with its remyelinating activity in cortical and in brainstem lesions. We next explored the value of these biological age measures at study baseline for predicting remyelination. In general, for two people with the same chronological age, an older biological age tended to be associated with less remyelination in response to bexarotene. However, these effects varied by region, and none were statistically significant in our population. For example, in brainstem lesions, having a biological age 1 standard deviation younger than expected for chronological age was estimated to increase the treatment effect on remyelination in the order of 30% for MRI brain age or 10% for blood biological age. In summary, this is the first demonstration to our knowledge that MRI brain age can be therapeutically modified by a drug in people with a neurological condition. Limitations of our study include the modest sample size and the post hoc use of existing data, which limited a more thorough multimodal assessment of biological aging. Strengths of the study include the ability to compare between blinded treatment and placebo arms, and the focus on assessing biological aging using data that would be routinely collected in clinical practice. Overall, this project highlights that beyond chronological age, biological aging may also influence the potential for brain repair in multiple sclerosis, and this should be considered when developing novel drug or lifestyle interventions.